This is a special episode of the Immunology Podcast, IUIS 2025, Day 2. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists in person. Today we're back with another special episode to discuss highlights from the IUIS 2025 Congress taking place in Vienna, Austria. If you're at the meeting, make sure to drop by the Immunology Podcast booth in the exhibit hall to meet the team and win some prizes. Today we'll be discussing some of the most interesting research we've seen presented over the past 24 hours of the meeting. So if you were in another session or weren't able to attend the meeting, we've got you covered. We're going to kick things off in just a minute, but before we get to that...
Are you studying natural killer cells? If so, make sure to visit STEMCELL Technologies booth to discover the new EasySep Human Pan-NK Cell Isolation Kit. Simply and efficiently isolate untouched, viable human NK cells from PBMCs or leukapheresis samples with EasySep. For more information, visit easysep.com.
So speaking of NK cells, did you have any NK cell talks you went to today, Brenda?
I did not, actually. They have been conspicuously absent from my talk lately. Did you have any NK cells?
I almost had an NK cell talk. So yeah, in the morning session on mechanisms of insight into cancer immunology, we had a soon-to-be guest coming who was there. So we had Eric Vivier on. He was who we recorded live today for a soon-to-be coming out episode, but he also had a talk, and he actually has a company that he spun off that specializes in antibodies that activate NK cells. So they will pull an NK cell to a target and then cause the NK cell to kill the target.
Okay.
But that wasn't what he talked about that day. He talked about antibody drug conjugates, which is why we almost got an NK cell talk, but didn't get an NK cell talk.
All right. Well, close enough.
But it was close.
Okay. What else did you see today?
Well, there was a lot of dendritic cells today for me.
All right.
So we had harnessing the immune system to prevent Lynch syndrome-associated cancers from Jolanda de Vries in the Netherlands. So Lynch syndrome is that mutation that people have that they're born with, a genetic disorder that makes you predisposed to cancers because you have mismatch repair gene deficits. So they'll get colon cancer, but all types of other cancers too. And then they die from it eventually. And they were looking at dendritic cell vaccines, which I think people are familiar with a little bit. You take immature DCs out, you add some antigen, you add something to cause it to mature, and you shove it back into people. And it's been kind of a weird story, right? It works really well in a few people and then doesn't work in most people. And it's driving people nuts because you think it really worked juicing those dendritic cells up, but it's been a problem. So early, the presenter talked about how early cases they've been able to get it to work in a patient with metastatic lung cancer, but it's just working in a minority of people. So they wanted to look at what you could do for Lynch syndrome because it's preventive, right? You want to give this to people early and see if they don't develop tumors. And what they had found as part of this process, and they're still trying to tweak this to make it really a therapy, but it's showing some promise, is that when they used a caspase-5 neoantigen, didn't work, but when they used the TGF-beta receptor 2 neoantigen, it was phenomenally successful.
Oh, okay. Is that a commonly generated neoantigen?
Not always, but the whole point was that when you had it, because there's all these random mutations all the time in the cancer, there's not just one neoantigen in Lynch syndrome, right? Because you're just getting random mismatch repair issues. But it worked well, and what they've realized is that that is a mutation that occurs early in tumor development, and TGF-beta suppresses proliferation. So the mutation makes you stop suppressing proliferation. So by creating a neoantigen to that, you stop the oncogenesis early in the process by targeting cells that have this escape for proliferation, because the mutations have to happen in a certain order for the cancer to develop. If you can get rid of this really awesome gene that stops metastasis, but if you're not proliferating already, your body's just going to kill it and it's going to die. So they are starting to realize that maybe some of these neoantigens for prevention or in general want to target early genes in the cancer neogenesis process that that need to happen early to get the cancer going. And so it prevents that from ever happening, so you don't ever get off the ground. So that was really cool. And then Maria Sibilia from Austria talked about myeloid cells and cancer immunotherapy. So we know EGFR myeloid cells are tumor promoting. That's been known. But what wasn't known is there's these EGFR therapies for CRC, for colorectal cancer. And everyone assumes it just works on the epithelial cells, right? Because epithelial growth factor on epithelial cells, you use an immunotherapy or an anti-EGFR drug on those epithelial cells, and boom, that's why it's working. But they showed through some knockouts that it's actually the myeloid cells really driving a lot of this. And what happens is you're targeting the EGFR positive myeloid cells, and you're getting rid of GMDSCs that are present when the EGFR positive myeloid cells are there, and getting rid of the immunosuppressive environment and pushing an immunostimulatory environment. And surprise, so PD-L1 drugs, right? They work only for mismatch repair CRC, not for most of it, but they do in their models when you add the EGFR therapy on board. And so they're starting to think about, you need the overriding of the myeloid suppressive part. So they're finding out this drug works differently than they thought and may have more broad use in cancer therapy. And then they also had a little talk about imiquimod therapy. Imiquimod's an immune potentiator on basal cell carcinoma, and that requires peripheral DCs, but doesn't work well in people. Generally known DC issue. But then they realized that mice groom themselves. And so when you put imiquimod on their skin, they lick it. And so they're actually ingesting some of it. So when they put a cone of shame, those cones of shame for on the animals, the protective effect of the benefit of imiquimod goes away. And so then they realized they can substitute interferon alpha instead of oral imiquimod. And then if you do that protocol in mice, it induces a TLR7 in macrophage. Remember the TLR from yesterday, and DCs, and that works now in melanoma and breast cancer models in mice. And so the next step is to go back into people with using interferon alpha as a potentiator, where imiquimod topically. So what you do is you put imiquimod on the skin cancer, good, but then you need to eat something for the potentiation. But instead of eating something, we'll just inject you with interferon alpha. That's the model, right? Is that the mice were eating the imiquimod. But you can replace that with interferon alpha. And the question is, does this now work in people? But because people can't eat imiquimod. In the same way. Well, you can, but it wasn't working in people. Like the imiquimod thing, like topical, sure, oral, It didn't. So they thought it was a DC thing, but now they're realizing maybe they just got to drug it right. But also I think it's cool that the whole effect from these mice was because the mice or other animals groom themselves. It's one of those wait a minute, they're eating it. Yeah. So I was deeply amused. And then of course, Eric Vivier's talk on ADCs. He's talked mostly about a Nectin-4 ADC that they're working on, because Nectin-4 is overexpressed in multiple cancers. This is known. The current Nectin-4 ADC that's out doesn't work particularly well, so they're redesigning something. And it was a talk about the company he has working on that. And all the cool stuff he's doing there.
So were the morning sessions, I guess.
That was one of them. That was the main morning session.
Okay. So my morning was focused on, I attended the session on epigenetic regulation of immunity. And I think, the two talks that I liked the most was the first one was from Johan Hun from the Helmholtz Institute for Infection Biology, I think it's in Germany. And Johan Hun has a really long, record on understanding epigenetic changes in regulatory T cells and characterizing the epigenome of regulatory T cells, and then he kind of been continuing this research. And so we all know that regulatory T cells have a very specific pattern of hypomethylation that changes the accessibility of certain genes, and that's part of the stable Treg signature, epigenetic signature. But then, so in this case, he started looking into the methylations of tissue infiltrating Tregs, or tissue resident Tregs, particularly the ones that live in the adipose tissue. And he showed a lot of data showing that there's these methylation patterns that are also characteristic of tissue resident regulatory T cells. And what's interesting is that there are differences between so not all tissue Tregs are the same, and that there are differences in, I think, methylation patterns and other things between regulatory T cells that are resident from different types of tissue, skin versus muscle versus adipose tissue, and that the stromal, so that part, to some extent, that these different signatures are driven by factors made by stromal cells from each tissue. And so that they have this differentiation protocol for making tissue-like regulatory T cells in vitro, and that one of the things they need is to add kind of stromal cells from each tissue for them to acquire the right resident phenotype. And what I think was really, really interesting, kind of the second part of his talk, is the influence of the microbiota on the development of visceral adipose tissue Tregs. Yeah, microbiota, Jason, yes. He's like, oh, microbiota. So he showed that germ-free mice have less VAT Tregs, visceral adipose tissue Tregs, also less colonic Tregs. So kind of the idea is to understand the differential influences. So it's not the same a colonic Treg and a VAT Treg, to show that they are different niches, have different responses to microbiota. So the question is, if germ-free mice have lower accumulation of VAT Tregs, right? What are the signals that are missing? And he shows that if you have certain kind of defined microbiota, this OMM12 is like a collection of different bacterial strains, and that is sufficient to restore the Treg population in the colon, but not in the vat. So again, indicating different niches require different signals. And so he tried to find out what is the signal that's missing in this particular microbiome composition. Basically, they show that there is that the production of indole metabolites, tryptophan metabolites, by specific microbiota species. I think this would be including this short filament. So I think actually, I don't remember if he said exactly which cell, which are the bacteria. No, he just used a bacterial strain that produced a specific enzyme, which was this tryptophanase, that would make this indole metabolites that activate the aryl hydrocarbon receptor, that in turn makes the stromal cells from the vat produce IL-33, and that IL-33 is the signal that the VAT Tregs are responding to, and that's kind of the order of events. And having a monoculture of bacteria expressing this tryptophanase was sufficient to restore the VAT Tregs. So Tregs not only in the vat, but also in the colon.
In the mice.
In the mice, yeah.
So interestingly, there is a consortium of bacteria that was in a phase 2 trial. A lot of them activate the aryl hydrocarbonase for IBD, and it just failed phase 2 for efficacy.
Interesting.
That we're finding a huge difficulty in translating designer vat-grown bugs with like, hey, this bug does this special thing. I'm going to mix it with 2 others that do these other things and get it to work as a drug. We think it would work like other drugs, and it's been a real issue. Yeah,
I guess that, in this case, they show that they have this bacterial strain, that they were looking for a bacterial strain that had this enzyme, and then they just made like a knockout of that enzyme. And then, I don't think they did not show it in collaboration with other.
Well, or even on its own, we found that like what works in mice and then what works in like an early phase in humans, like once you go to the bigger stuff, it's shockingly failing. It's like, as of like this last week or two, the field's like, well, what about cultivated products? The donor derived are different because it's more of a shotgun approach, right? But like for the ones that like, hey, I want a bunch of bacteria that do A and or other ones together, it's become a real issue. So I think this is super interesting. But now I'm wondering like, oh, you just said the pathway they couldn't get to work.
Yeah.
It's interesting. Like I want it to work super bad, obviously, and like now it's sad panda.
Yeah. I mean, it works in mice. Okay.
You know how much cancer we would cure in mice? Mice in an alternate world where all the research is used for mice,
Of course, this is really also these are, there's also there's things they do it in male mice because they have more fat, they have more adipose tissue to get cells from. They say that so mostly it's easier to look the effect in older mice because as they get older, the differences in the development of this population become more and more apparent. So it's not an easy thing to measure, and I'm sure there's other effects that are hard to control for. But continuing on the topic of epigenetic regulation, there's also a talk from Wilfried Ellemeyer from the University of Vienna, and he was looking at the role of histone deacetylases in regulation of T-cell mediated immunity. And basically, he spent some time talking about the importance of lysine acetylation by acetylases. And what's interesting is that, well, of course, we all know histone acetylases and deacetylases and their effect on epigenetics or altering histones, and therefore the accessibility of certain genes for transcription. But it's also important to keep in mind that the histone the acetylases and acetyltransferases and deacetylases have also many other targets besides histones. And like he mentioned, over 20,000 sites in over 8,000 proteins. And the acetylation is a type of post-transcriptional modification that affects the function of many different proteins. And so he looked into a model of EAE in autoimmunity, autoimmunity model EAE, and what happens if you are knocking out histone deacetylase 1, HDAC1, and basically by knocking out HDAC1, there's a protection from EAE, and this has also been shown that HDAC, and so HDAC inhibitors can ameliorate the clinical score of EAE. And one of the things that they show is that, to some extent, this rescue of EAE happens through diminished cell migration because of the downregulation of certain integrins and adhesion molecules as a consequence of this inhibition. And they kind of checked that it really depended on the acetylase function of HDAC1. And because they did this, because sometimes there's more than one, there's HDAC1, HDAC2, and if you knock out the protein, you might be having the HDAC2, for example, take over, or you might have other interactions of this protein that get also disrupted. So they show that if they actually remove the catalytic part of this protein, and so preventing the acetylase function, that is sufficient to mediate this abrogation of EAE. So it really is this HDAC1 specifically is what is mediating the effect in amelioration of clinical EAE.
Yeah. So in postdoc, the lab I was in mostly studied a protein family that when you knocked it out, disrupted chemotaxis of leukocytes. Mm-hmm. And its primarily was found from a screen in EAE.
Right.
No leukocyte migration, no EAE.
Yeah. Yeah. So really clearly very important for that. And yeah, so that's kind of my morning. And then I guess we both have the Dan Lidman's plenary talk.
Yeah, which I think covered a lot of what we talked about him with. Didn't you think? We had him on.
Yeah. So basically he talked about the role of orgametie, a little bit of a story of orgametie. It was really interesting. And yeah, but I think some of his stuff was new. So the effect of orgametie. So he introduced to these orgametie positive APCs that now his postdoc, the postdoc in his lab discovered published a few years ago. But so that was the story originally was on like early development and the early waves of T-Rex and early development of T-Rex. But also, I think there was a really nice data on the effect of on really pinpointing and characterizing the cells and other transcription factors besides orgametie that are important for them. I think the role of PRDM16 on these antigen-presenting cells, that the fact that they seem to be of a dendritic cell lineage, which was not clear because at the point when these cells were first described and people were kind of putting them together with ILC3s, or people were making them. It was unclear what was their
It sounds like some of the
lineage.
ROR gamma T positive ILC3 story is really a dendritic cell story. And so some of that old data may need to get revisited in that lens. Also, then you looked at immune tolerance or oral tolerance, right? And so some of the different things that really the relative to the antigen that you swallow, whether it's food or bacteria, what drives oral tolerance downstream, and because it's different cells and different genes for different stuff, which was cool. But yeah, again, today's a DC day.
Yeah. Dendritic cells are very important.
Yep. And then I went to a skin and barrier immunity talk, which was really cool. And so this was, I didn't get to see the whole session. I got to see one. And this is Vanderlei da Silva Fraja, who junior from Brazil. And he was looking at inhibition of a pathway called 11B-HSD1. But long and short, we know that corticosteroids inhibit wound healing. And it's worse in diabetic mice. And so he was able to find, he had a specific drug called metatripopone. These names are not meant to be spoken, as we know. And it inhibits part of the pathway for glucocorticoids, dropping their levels. And he was able to show in a wound system that you have increased collagen deposition through myofibroblast differentiation, and it reduced the glucocorticoids at the site directly. And so that was cool. And then there was another talk by Fabian Stritt from Germany, and he used an immunomodulator S-soluble CD83, and that inhibited a wound healing model of pressure ulcer. Now, you can't make mice sit still to give them a pressure ulcer like you get from sitting in a bed for a long time. So they use magnets on the back skin of the mice to cause ischemia and then release it to cause reperfusion. And so they were showing that this drug had better architecture restoration, and they were able to track the timeline down. So it changes by day 8, cellular change, transcription changes by day 8, and cellular changes by day 12. So those are pretty neat. And then I went to some poster sessions, and I am now getting a picture of a poster just because, sometimes posters are the unsung heroes. And so I have to give a shout out to Jay Jimenez Loyoza from Australia and her poster looking at specific bacterial species and their ability to immunomodulate immune cells by limiting STAT3 signaling. So she was able to find that supernatant from a subset of bacteria they're kind of standard healthy bacteria. So these are some Bacteroides species and some Clostridium species are able to stop overexpression of STAT3 activation. And you don't need the bacteria, you can just do the supernatant, which is pretty cool. So that was earlier work, but they were using, a cell system and stuff. But, giving shout out because posters never get the love. Yeah. And a lot of the early trainees are in the poster mode. Posters. And there are lots of posters, and I went to them and had some fun. And, shout out to someone I got to talk to.
Nice. That's good. I didn't make time to go to the posters, unfortunately. Maybe next session.
There's some more tomorrow.
Yeah, there will be posters every day. All right. So I think just before we go, I just want to also just shout out to the 3 rising stars that presented this time after the plenary session from Dan Littman. So we had Thomas Krausgruber from the CEM from Vienna, and he was talking about the importance of the differences in the response of structural cells after infection with strains from LCMV that were either the Armstrong acute infection or the clone 13 chronic infection, and how that has to do with the differences that we see in the immune response to these 2 viral strains. And Thomas Volkel from the Medical University of Vienna was a very interesting talk about the longitudinal studies looking at the development of immune responses, and especially how in early life, in the first year of life, there's this massive changes in the repertoires of antibody responses and the specificity of these antibodies and the effect of, for example, cesarean section in the repertoire of babies, kind of even later in life. And we had also Ranit Kenmi from, we've been discussing kind of similar to some of the work she did, talking about the the effect of this ROR gamma T positive dendritic cells in the induction of immune responses and regulatory T cells, and how they will suppress disease activating inflammatory cells. So it was really nice session as well. So
more DCs.
Yeah, dendritic cells.
All right, today's theme. Every day has a theme. Today's theme is dendritic cells.
Indeed, they are. They're running the show, aren't they, in the end?
Yeah.
All right.
Well, that brings us to the end of our second IUIS 2025 episode, Revenge of the DC. Don't forget to follow us on X at Immunopodcast or Blue Sky at immunologypodcast.com to find out what we're up to at the meeting. And visit us if you're here at the Immunology Podcast booth on the exhibitor floor, where you can enter for a chance to win a gift basket. Check back here tomorrow for another episode recapping day 3 of the meeting. See you then.